Hi, welcome to bioinformatics classes. Today I am going to give the introduction about bioinformatics databases. What is a database? A database is a convenient system to properly store, search and retrieve any type of data. A database helps to easily to handle and share large amount of data and supports large scale analysis by easy access and data updating. Biological data, dynamic and multidimensional. Modern high throughput techniques output huge data, and there is a great need to collect and store the data in organized format. Here, properly, if you see the database conveniently, to store the DNA sequence, protein sequences, and to retrieve the literature, we need the databases, which actually to easily to acquire the information of a particular experiment or the particular gene information, particular proteins. So the database is very important. So the biological data sequence, genome sequencing of different organisms. This is the biological data representations. You see the genome sequencing, how the genes are sequenced and the information is stored here. The more the information is stored here is the reptiles. The biological data, the presentation of the bar representing showing the biological data storage. What are actually biological databases? Biological databases are the collection of libraries of a biological information which is retrieved from scientific experiments, published research articles, high throughput experiment technologies, and computational analysis. The information you may get from the DNA, proteins, gene expression analysis. If you get the information from the DNA, it's called genomics, and then protein is the proteomic studies, microarray gene expressions. The informations contained in biological database includes gene function, structure, localization both in the cellular and chromosomal level, biological sequence and the structures. If we see the central dogma of a molecular biology, the databases contain DNA information, RNA information, and the protein. The DNA information is a genomic DNA database, cDNA libraries, protein sequence database. The National Center for Bioinformatics Information (NCBI) is the large database. It stores all kinds of informations. Here we see the NCBI homepage. We submit the DNA sequence. We also retrieve the information. We download the DNA sequence informations. All resources we can find from the NCBI website. Chemical assays, bioassays, scientific literatures analysis data also we can retrieve from the NCBI. RNA sequence, DNA sequence analysis, the domain structure, protein domains, and the structure analysis, the protein sequences, gene expression analysis, and genetic information, and the genomes and the maps, homology, literature, the protein sequence. It is the biggest databases used in bioinformatics. GenBank. This is the database where you get the information regarding the genetic or the DNA sequence information. GenBank is the genetic sequence database at the NCBI. It was established in 1982 and is maintained by NCBI. The gene sequences, DNA sequences, first initially submitted to GenBank using the several different methods. It contains publicly available nucleotide sequence for more than 240,000 immunoorganisms, obtained primarily through the submission from individual libraries and batch submission from large-scale sequence projects. GenBank records. The GenBank records the database stores header information, feature information, and sequences. Header shows the information that apply the whole record of the gene and the annotation on the records. Sequences. GenBank record header information. So these are the information where the gene sequence is obtained in GenBank databases. Locus, definition, accession, version, keywords. The definition is a Homo sapiens. If you see there is the GenBank information of a particular gene, this is 628 base pair mRNA. A source is the Homo sapiens organisms, and the similarities you can find the gene similarities in eukaryotic metazoans chordate, all primary reference and the author's information who worked on this gene sequence, title where the gene sequence is obtained, the title of the research publication and where it is published, and submission ID PubMed. Gene informations and the reference and then other authors who also worked on the same gene sequence. GenBank records features. What is the feature? Information is the source where it is obtained the gene and the gene abbreviations, exons and the misfeatures and cDNAs. And then the GenBank record in this sequence analysis. If you see here the gene sequence of DNA, which is 601 base pairs. This is the format of the gene sequence. The versatile compact one-hitter NCBI reference sequence, GenBank sequence, FASTA sequence. Homo sapien hemoglobin subunit beta HBP mRNA sequence. Accession numbers are labels for the sequence. NCBIs includes the databases such as GenBank that contains information on DNA, RNA, protein sequence. So for the we submit the DNA, we get the accession number. For the RNA, the other accession number. The protein or protein sequences. You may want to acquire information beginning with the queries such as the name of the protein of interest or the raw nucleotide comparing a DNA sequence of an interest. DNA sequence and other molecular data are tagged with the accession numbers that are used to identify the sequence or other record relevant to the molecular data. The reference sequence, database of the reference sequences, curated, non-redundant, one record for each gene or each splice variant from each organism represented. Each record is intended to present an encapsulation of the current understanding of a gene or a protein similar to the review article. So ensemble is a genomic databases. Same as an ensemble, the information you get all ensemble, you will get the blaster sequences, tools and different sequence analysis. UCSC Genome Browser, a genome visualizing database. The other genomic databases are online Mendelian inheritance in man, OMI. You get the information from these databases regarding the particular gene. Sequence Read Archive, SRA, database of genomic variant, gene expression omnibus, AG biodata, Arab data's information resources. The effective strategies for the various biological databases search. See the difference of 1 and 2, 1 or 2, 1 and not 2. So different databases search the different analysis. Biological sequence analysis. Why we compare the sequence? To identify the genes, determine the function of each gene, identify the proteins involved in the regulation of gene expression, to identify the sequence repeats, identify the other functional regions, for example of origin of replication, Pseudogenes, sequence of responsible for the compact folding of DNA, and sequence responsible for nuclear anchoring of DNA. Similar genes arise by gene duplications. Similar genes arise by gene duplication. Copy of a gene inserted next to the original. Two copies mutated independently. Each can take on a separate functions. All or a part can be transferred from one part of genome to the another. Sequence comparison method. The sequence comparison methods work for dot matrix analysis, dynamic program, word archetypal method, blast analysis, the progressive alignment methods. So these are the four programming methods and involves in the sequence comparison. Pairwise sequence alignment. Pairwise sequence alignment, fundamental operation of the bioinformatics. Actually, it is used to decide that if two proteins or genes are structurally related or functionally. Either they are structurally similar or functionally. It is used to identify domains or the motifs that are shared between the proteins. Pairwise alignment or the sequence alignment helps in identifying the domains and the motifs which are in the protein. It is base of the blast searching. It is used in analysis of genomes. Pairwise alignment protein sequence can be more informative than the DNA. The pairwise animals we will get a lot of information than the DNA sequence similarity search. A protein is more informative. Many amino acids share the relative biophysical properties. The codons are degenerative, change in third position often and do not alter the remaining amino acid that is specific. DNA sequence can be translated into protein and then used in the pairwise alignment. But not always. Many times DNA elements are appropriate to confirm the identity of cDNA, to study non-coding region of DNA, to study DNA polymerization, example Netherlands versus modern human DNA. So next class we are going to continue